Hi, I'm Vicki Fang, and I did my PhD work in the lab of Susan Schwab at NYU. Today I'll tell you about my work about how S1P gradients in lymph nodes regulate natural killer cell positioning and infection response. So the immune system detects and fights off foreign particles, like bacteria, viruses, parasites, as well as cancers. And because you can get these pathogens and cancers anywhere in your body, the cells of your immune system are special in that they need to move around your body in precise ways to protect you while maintaining host fitness. Now, humans have hundreds of lymph nodes, which are these little organs all over your body, packed full of lymphocytes, primarily T cells and B cells, but also smaller populations of innate cells like natural killer cells. And lymph fluid collects material from all of these other tissues of your body and then drains all of that into the nearest lymph node, where foreign pathogens and antigens from these other tissues can be detected. So how does S1P play a role in these immune cell movements? I'll use T cells to illustrate what we know about this. So T cells in a lymph node move around constantly searching for their cognate antigen. If they don't find it, that T cell can leave the lymph node into the lymph, meet up with the blood, and eventually find its way into another lymph node where again the T cell will look around for its cognate antigen. Now S1P is a lipid chemoattractant molecule that is higher in concentration in lymph compared to lymph nodes, and it's this gradient that guides naive T cells out of the lymph node into the lymph fluid. We know that spinster2 is the S1P transporter that generates these gradients at homeostasis. What we don't know is what these gradients look like inside lymph nodes themselves and how these gradients may influence an immune response. It turns out that besides effective migration around your body, even with these small lymph nodes, cell movement has to be coordinated in precise ways to mount an effective immune response. In this talk, I'd like to tell you about how S1P gradients inform natural killer cell migration that happens inside the lymph node and why this can be important during infections. To start, I'll tell you about lymph nodes themselves. So they're these little organs, again, that drain all of the other tissues like the lung or the skin, and they're an important meeting place for various immune cells to get together and get activated to fight infection. Now, lymph nodes are a highly organized tissue. So B cells reside in B follicles around the lymph node. T cells are primarily in the center of the lymph node, and important for my talk are these blue vessels sitting at the edges of the lymph node, and pathogens from the skin or the lung, for example, can drain quickly through the afferent lymphatics, get to the lymph node in the subcapsular sinus, and then quickly this fluid can drain into the medulla, where all this lymph fluid gathers before leaving via the efferent lymphatics. And it's important for the lymph node to have defenses against lymph-borne bacteria so that these pathogens can't escape the lymph node through these efferent lymphatics and become a more systemic infection. Now, when a pathogen enters, how does the lymph node protect itself? So there are actually macrophages, shown here as these green squiggly cells, as well as NK cells, these brown cells, that are sitting at the periphery of the lymph node. And when a pathogen comes in, the macrophage gets activated very rapidly and secretes a cytokine called IL-18. This IL-18 signals to the nearby NK cells that then get activated and secrete their own cytokine, interferon gamma. It's this interferon gamma that can communicate with macrophages to upregulate microbicidal activity and thereby help control the pathogens within the lymph node. Now, for this process to be effective, it seems important that number one, the macrophages and the NK cells are pretty close together so that they can communicate well. And number two, that they sit near the periphery of the lymph node where these pathogens are coming in so that they're ready to sense and respond to them. But it was unknown what factors are required for the peripheral positioning of these cell types at the periphery of the lymph node. And we hypothesized that maybe it was lymphatic endothelial cells that are secreting some sort of a chemoattractant in that area to bring immune cells to the lymph node periphery. And we thought that that chemoattractant might be sphingosine 1-phosphate. But actually, it's very hard to determine where the S1P is within tissues. And that's because the locations of signaling lipids in vivo in general are pretty difficult to define. Number one, lipids are not genetically encoded, so you can't tag them with a GFP or other fluorescent marker. Number two, Shown on this slide is a busy illustration just to show you that there are so many enzymes involved in the production, the degradation, and transport of lipids like S1P that you can't possibly predict S1P levels simply by enzyme expression levels. And finally, mass spec is infeasible since many of the lipids we're interested in are made intracellularly by all cells, but we're actually interested in the extracellular signaling fraction that can act as a chemoattractant. So if you measured S1P in a lymph node, it would give you artificially high values of S1P due to detection of intracellular S1P. So to get around this problem, a previous grad student, Willie Ramos, generated a reporter mouse based on the concept that S1P receptor 1 gets very sensitively internalized when it binds S1P. And the way this reporter mouse works is it has two fluorescently tagged proteins split by a P2A skip sequence so that they will be transcribed and translated one to one. So one is an S1P receptor 1 tagged with GFP, and this receptor, remember, will get internalized when it binds S1P. The other is a non-internalizable, non-signaling form of human CD2 protein tagged with RFP. So the human CD2 RFP will sit on all the cell membranes. And so you can imagine if you're a reporter cell sitting in an area of low S1P, shown here on the left, then the GFP S1P receptor 1 construct will stay on the cell surface of the membrane along with the RFP protein, and what you'll see is a yellowish green cell membrane. However, a reporter cell sitting in an area of high S1P, shown here on the right, is sitting in an area of high S1P that's going to cause internalization of the S1P receptor 1 GFP protein. And so a cell membrane that's sitting in an area of high S1P will therefore have orangey or redder membranes. So we were essentially able to generate a heat map of S1P in tissues, where orange and red membranes indicate hot or high levels of S1P, and greener membranes indicate cooler or lower levels of S1P. And that the S1P levels are going to inversely correlate with the GFP to RFP ratio on the cell surface. So this is a reporter lymph node section from a control mouse. Zooming in on the areas in the medulla and the T-zone revealed very clear gradients within the lymph node. So these cells are green low in the T-zone and then relatively red high in the medulla, which is at the periphery of the lymph node. And notably, at the subcapsular sinus, these cells are very red and high, and that's because the subcapsular sinus is carrying lymph fluid, which is known to be rich in S1P. And S1P levels in these regions were quantified as the ratio of GFP to RFP on the surface of reporter cells. And again, higher ratios indicate lower levels of S1P. We did verify the fidelity of the reporter in several ways that I don't have time to show here. But we next wondered, how might we disrupt this gradient and what's required to generate this gradient? And what we found is when we transferred bone marrow from reporter mice into irradiated host mice, where Spinster2 could be deleted by an inducible Krey-Lox system, we were able to see what the reporting would be like in a Spinster2 deficient mouse. Remember, Spinster2 is a transporter of S1P expressed on lymphatic endothelial cells. And you can see here that in the absence of Spinster2, the gradient between the T-zone and the medulla is now flattened, and quantification is shown on the right side. Our next question is, are these gradients important for cell localization? What are the function of these gradients? So we looked at lymph node organization in Spinster2 deficient mice, again, where lymph node gradients are disrupted. And I'm not showing the macrophages here, but they were largely fine. NK cells, however, stained or shown in green here, look mislocalized from the medulla into the T-zone. And we quantified this in various ways, all of which showed a relative increase of NK cells in the T-zone and a decrease in the medulla. This is the ratio of the NK cell density in the T-zone compared to the medulla, and you can see that in Spinster deficient mice, this ratio is a lot higher. We next asked, how are NK cells sensing the Spinster2 generated S1P gradients in lymph nodes? Now there are 5 known S1P receptors 1 through 5, and NK cells express high levels of S1P receptor 5. And they're known to use S1P receptor 5 to egress from lymph nodes. So we asked whether S1P5 might allow NK cells to also position themselves properly within lymph nodes. When we knocked out S1P receptor 5, the animal showed the same phenotype as before, that is to say, mislocalization of the NK cells from the medulla to the T-zone, And again, the quantification is to the right. We did generate mixed bone marrow chimeras with these S1P5 knockouts and wild-type NK cells within the same wild-type host. And this demonstrated that S1P receptor 5 was required on NK cells in a cell-intrinsic manner. One thing I'll note here is that although NK cells are no longer preferentially in the medulla when we disrupt S1P gradients or the signaling, not all of the NK cells left the medulla. You can still see many NK cells localized in the medulla. And so this suggested to us that another unknown player might also help NK cells maintain localization in the medulla. And so when we were thinking about what this other player might be, we thought about previous work. So we knew that Jason Sister's lab had previously shown that the chemokine CXCL12 was abundant in lymph node medullary cords. Shown here are the green GFP, CXCL12 GFP, and again you can see that it's very abundant near the medulla of the lymph node. So we investigated whether CXCL12 and its chemokine receptor CXCR4 might also contribute to NK cell positioning in the medulla. First, we verified that lymph node NK cells expressed high levels of CXCR4 by flow cytometry. So CXCR4 expression here is on the x-axis, and you can see that NK cells stain much more brightly for CXCR4 than the CD4 cells within the same lymph node. So we treated mice with a drug called AMD 3100. It's a CXCR4 specific antagonist. And again, we saw mislocalization of the cells, of the NK cells from the medulla to the T-zone. And we noticed that hitting both receptors resulted in a slightly more mislocalization than hitting either receptor alone. And here's the quantification, again, for all four conditions. Now, so far, what I've shown you is that, number one, we used the reporter mouse to map S1P gradients, and we saw that these gradients were higher in the medulla compared to the T-zone, and that this gradient is generated by the transporter Spinster2. Number two, I showed you that these gradients are sensed by S1P receptor 5 on NK cells, allowing NK cells to localize to the medulla of lymph nodes at homeostasis. And finally, I've shown you that CXCL12 and its player CXCR4 is also required for NK cell positioning in the lymph node medulla. So our final question was, why does NK cell positioning matter? To test this, we infected control and Spinster2 deficient mouse foot pads with Salmonella typhimurium, and then we harvested their draining lymph nodes about 2 hours later. In Spinster2 deficient mice, NK cells made interferon gamma at about half the frequency as their littermate control counterparts. But to really nail down whether this defect was a localization problem or whether NK cells in these mice were defective for some other reason, we stained draining lymph node sections for NK cells and interferon gamma. And we saw that NK cells in the medulla, whether you're in a Spinster2 deficient mouse or a control mouse, are able to make interferon gamma equally well, quantified at the right. However, NK cells in the T-zone, in again either the deficient mouse or the control mouse, was not able to make any interferon gamma. So these experiments showed that localization was really important for NK cell responses to infection. We also replicated these experiments in both S1P receptor 5 knockouts, as well as the mice treated with the AMD 3100 that blocks CXCR4. And we saw similar results. So again, in control mice, an average of about 20% of draining lymph node NK cells are secreting interferon gamma 2 hours after foot pad infection. But in S1P receptor 5 knockouts, in AMD 3100 treated animals, and then the dual inhibited animals all have decreased frequency of interferon gamma secretion. Now finally, we wanted to measure the effect on bacterial control. The diminished secretion of interferon gamma by NK cells near the medullary sinuses should impair the ability of nearby macrophages to contain infection within the draining lymph nodes. This should cause the infection to be able to spread downstream to further lymph nodes. So here, in AMD 3100 treated mice, which again blocks CXCR4, we measured the CFU ratios of the downstream iliac lymph nodes compared to the primary draining popliteal lymph nodes. And you can see that there's a slightly higher CFU ratio in AMD 3100 treated mice compared to control mice, suggesting that positioning of NK cells in the medulla does help bacterial containment within draining lymph nodes. So to summarize, prior to our work, we knew that lymph node parenchyma likely had lower levels of S1P compared to lymph fluid, and that allows lymphocyte egress from lymph nodes. But we were able to visualize S1P gradients within the parenchyma, and we saw that there are high medullary gradients of S1P generated by spinster2, and we also learned that CXCL12 and CXCR4 signaling within the medulla is important for a lot of these NK cells to position themselves properly in the medulla. Now, more generally, the lymph node periphery is increasingly recognized as an important site of many immunological functions, from pathogen containment to helper T cell differentiation and innate immune cell responses. It seems that there may be spatial territories for different functions of the lymph node to mediate responses to various infections, insults, etc. And more work needs to be done to understand these tissue territories. And finally, I'd like to thank members of the Schwab Lab for discussions and collaboration, Ken Cadwell for the Salmonella enterica typhimurium, Ken again, Joel Ernst, Adrian Erlbacher, and Dan Littman for reagents and helpful discussions, the NYU microscopy and cytometry cores throughout this project. We also got assistance from the molecular cytology core at Sloan Kettering for microscopy and image analysis. We got the NIBR reagent from Mark Bagad and Volker Brinkman at Novartis, and we got the S1P receptor 5 knockout mice from Gerald Chun and Richard Rivera. And we would thank Bea Breyer and Jason Sister for critical reading of the manuscript, and the funding is shown here. Thanks so much.